Hello, dear students. Today we're going to speak about chemical nature of genes. Let's start. A capsule is an outer coat covering a bacterial cell. Unlike a slime layer, it is not easily washed off. Although capsules are not essential for bacterial growth and reproduction in laboratory conditions, they can make the difference between life and death in natural situations. For example, Streptococcus pneumonia has non-capsulated and capsulated strains. Those lacking a capsule are easily destroyed by the host and do not cause disease. However, the capsulated strain kills mice quickly. The capsule helps the bacterium resist phagocytosis by host cells. It contains a great deal of water, protecting the bacterium from desiccation. It keeps out detergents which could destroy the cell surface membrane, and it helps bacteria attach to host cells. We know today that DNA is a chemical in which information is from parent to offspring. This spread looks at how researchers established this link between DNA and inheritance. In the 1860s, nearly 100 years before Watson and Crick's work on the structure of DNA, Gregor Mendel established that inheritance depends on factors that are transmitted from parents to offspring. In 1909, it was found that patterns of inheritance were reflected in the behavior of chromosomes. Wilhelm Johannsen referred to these factors as genes. Genes were assumed to be located on the chromosomes because genes that are inherited together were found to be carried on the same chromosome. However, the chemical composition of genes was not known. Protein or DNA. Which is genetic material? Chromosomes were known to contain both protein and DNA. Most biologists assumed that proteins with their highly complex and infinitely variable structure were the inherited material. The nucleic acids were thought to be too simple to carry complex genetic information. This view was reinforced by the work of Phoebus Aaron Levine. Levine made major contributions to the chemistry of nucleic acids, but believed mistakenly that DNA was a very small molecule, probably only 4 nucleotides long. In 1928, Fred Griffiths, an English medical bacteriologist, published a paper describing experiments on pneumococci. His results set the stage for the research that finally showed that DNA is the genetic material. Griffiths' experiment. Transformation of pneumococci. Pneumococci are bacteria that cause pneumonia. They occur in 2 strains, a disease-causing smooth strain, strain S, and a harmless rough strain, strain R. Strain S has a capsule on its cell surface. This capsule is absent from the harmless strain R. Griffiths found that mice injected with live strain S soon died, but those injected with live strain R survived. Mice injected with dead strain S bacteria all survived. The results of this series of experiments were as expected. However, the results of Griffiths' next series of experiments were thoroughly baffling. Mice injected with a mixture of heat-killed strain S and live strain R died. Moreover, Griffiths recovered live strain S bacteria from the dead mice. After many careful experiments, Griffiths concluded that hereditary material had passed from the dead bacteria to the live bacteria. This changed harmless strain R bacteria into virulent strain S pathogens. This process is called transformation. Avery's experiment. DNA was a transforming agent. In the 1940s, Oswald Avery, Colin MacLeod, and Macklin McCarthy showed that DNA was responsible for transformation. They used enzymes that hydrolyzed polysaccharide, DNA, RNA, and protein on samples of the disease-causing strain S pneumococci. Different samples had different parts of their cells destroyed by these enzymes. The researchers then exposed strain R pneumococci to the treated samples of strain S. The transformation of strain R to strain S was blocked only when the DNA in the sample was destroyed. These results provided strong evidence that DNA carried genetic information for transformation. However, many scientists remained unconvinced. In 1952, Alfred Hershey and Martha Chase performed several experiments with T2 bacteriophage, a virus that infects bacteria. Their results convinced even the skeptics that DNA and not protein was the genetic material. Electron micrographs indicate that T2 bacteriophage infects Escherichia coli by injecting its DNA into the bacterium while leaving its protein coat on the outside. The phage takes over the genetic machinery of the host cell to make new phages. Eventually, the bacterial cell bursts, releasing new phages to infect other bacteria. That is the end of our video session. Next time we're going to speak about chromosomes. Thank you for watching.